Hello, biologists. Coming to you live today from my backyard in January, where it is 65 degrees. Crazy. Okay, we are going to go from just replicating our DNA to actually the genetic code. We're going to unravel some DNA and we're going to make a protein. Okay, so we're going to talk about the genetic code and we're going to compare and contrast prokaryotic and eukaryotic transcription and then translation. So let's dive into 15.1. We're going to start with what's called the central dogma. This is talked about all the time, and it sounds a lot more detailed than it is. When someone says the central dogma of DNA, basically what that means is that we're going from DNA to RNA to amino acids and then proteins. DNA, RNA, proteins. That's what the central dogma means. We're going to get into the details of that, but that's what it means. And then we're going to talk about how the genetic code actually translates into amino acids, which leads to proteins. Okay, starting off strong with mRNA, messenger RNA. Okay, so essentially we are going to make a copy of the DNA and we're going to make RNA out of it, right? Instead of A, T, C, and G, we're going to have A, C, G, and U. No T, no thymine. We're going to switch that out for uracil. Okay, that's how you know it's mRNA, because it's got a U in it. It's got a uracil in it. We're going to make an mRNA template, and we're going to move that out of the nucleus so that it can get translated. So the first thing we're going to do is transcribe. If you remember how I think about this is if I ask you to transcribe my notes, you're going to put it still in English. So yeah, there's a U, but the transcription is just making a template. So that's what we're starting with. That's what the mRNA does. It transcribes the DNA into RNA so it can leave the nucleus and begin the process of making a protein. So protein synthesis consists of 20 commonly occurring amino acids. So our mRNA will code for a amino acid, which will make ultimately a protein. The different amino acids have different chemistries and different structures, which we're going to talk about a little bit, and that is going to make the various proteins. So the flow of genetic information, again, this is the central dogma, starts with your DNA. That's going to transcribe into mRNA, messenger RNA. That would be the postcard or the piece of paper. If I said transcribe my notes, I would want you to give me a piece of paper of those notes. That's the mRNA. Then this will move and decode into, right? So here's your mRNA transcript. There's going to be some introns and exons in there. Those are going to be, the introns are going to be cut out because they stay in. The exons exit. The introns stay in. That's going to be all packaged up, right? When we have the RNA, we have all these things that get transcribed, the transcript, and then somebody is sort of going to edit it. Right? Maybe you transcribe my notes and somebody's like, listen, Gullivi talks too much. We're going to cut out the stuff she says it doesn't matter. And we're going to cut and paste that into like a very nice set of notes. And we're going to put like a heading and a like sincerely. That's what there's going to be a cap and a tail and it's going to prepare it all really nice. Put it in an envelope and then move it into the ribosome so it can be translated. So that is the essential overview of the central dogma. You're going to make mRNA, then you're going to process it, make it all neat. You're going to make it neat. You're going to leave the introns because they stay in. You're going to cut those out. Make sure you just have exons. You're going to put a poly A tail. You're going to put a cap on there. You're going to put it all in the envelope, and then you're going to send it into the ribosome to make this polypeptide chain. The copying of the DNA to RNA is pretty straightforward. One nucleotide is added at a time for each RNA. For each DNA, there's going to be an RNA. We only do one at a time. The translation to protein is a bit more complex. We will get to that. One of the things that is sort of just a vocabulary word that you need to know is the word collinear, meaning that this is going to go along in the exons only. And we're going to read 3 at a time, right? We're going to read 1 through 3 will correspond to an amino acid. 4 through 6 will correspond to amino acid. Codons, groups of 3, will code for each amino acid. And that definition is called collinear. every 3 nucleotides, codons, code for one specific amino acid. AUG, for example, GUC, CUA, it codes for methionine, valine, leucine. This is a direct collinear relationship. We are going to transcribe and translate some mRNA and dRNA in class. You're going to get this delightful why is this being so weird? Hold on. I do not know. Hold on. Sorry. So you are going to get this amino acid code. And what you are going to be able to do, we will do this in class several times, is I'm going to give you a DNA. And then you're going to transcribe it into an mRNA. Then you're going to look at this code. Let's say your mRNA is CUU. That's going to code for leucine. Okay? One of the words that we need, again, in our vocabulary is degenerate. Now, typically, that is not a positive word. If I called you a degenerate, you would take offense to that, which is fine. But in this case, it's not like a negative, like stupid degenerate code. Essentially, it means a given amino acid could be coded by more than one nucleotide. So let's take a minute and look at this. Like, let's look at arginine here. Look at this. And even down here. Look at all of the ways you can get an arginine. You can get a CGU. Really, if you have C and G, pretty much any other third position in the codon is going to give you arginine. Now, let's just take a minute and think deeply about that. That is going to really help when we get to mutations. Really, you just need the first two, and you're going to get the correct protein, and that is important. Some of the rest of these you can see there are several. Look at all the ways you can get arginine here. Glycine is the same. 4 options. You just really need a GG, and then you're going to get glycine. Now, some that we need to be a little bit more serious about. AUG, only methionine. There's only one code that's going to be the start codon, which is methionine, because we don't want to be messing around with that and just start translating things willy-nilly. There's 3 stop codons. So this is what it means when we say degenerate. It's not a like slang negative term. Yeah, degenerate. It means that there are 3 codons, but sometimes only the first 2 will get you, like with arginine. You just need a C and a G, and then no matter what this third position is, you get arginine. Or in the case of leucine, you need a UU, and then if you have an A or a G, you get leucine. Or if you have a CU, all of these give you leucine. Okay, so Francis Crick and Sidney Brenner used a chemical mutagen to determine this because it was not obvious, right? They knew that you have the mRNA and it is somehow coding for an amino acid. They did not know how that was happening, nor did they know how many letters you need to make a word. Does that make sense? So is there 2? Is there just like UU? Is it 1 for 1? Like every nucleotide codes for an amino acid? Is it 4? Every 4 nucleotides code for an amino acid? So there were several scientists working on this. There were several experiments being done. And essentially, when they used 1 or 2 nucleotides to try and make the amino acid connection, proteins were not produced. But when 3 nucleotides were inserted, the protein was synthesized and functional. So that is the overall experiment that Francis Crick and Sidney Brenner did on proflavin to determine that the coding was 3 nucleotides. And as we look into mutations, this will be more and more important, and we will talk about different types of mutations. One of the things that we do need to talk about is when you think of mutations, what is going to be the worst? Well, as we talked about, as we set up here, right? If you get UUU, sometimes you get what's called a silent mutation. So let's go back to this one and say, here we have serine, valine, right? These 3 are serine, these 3 are valine, right? Maybe you have CC. Let's see what should goes for valine. Let's go back up here. Where's our, right? All you need is a GU. Where else is valine? I feel like it's got to be somewhere else. Okay. So if you have GU, you get valine. What do we have here? We have a GU. And we get valine. But here, now we have a GC. And you get a completely different, or you've actually taken this out. Now you're going to get the whole thing is shifted. But if you went GUU, silent mutation, it's not going to change anything. So you can see moving forward, I want you to think in your head how this is going to translate to what kind of mutations are going to cause what. One letter right? Sickle cell anemia is just a one letter difference. Just one of these letters is different and that causes sickle cell. A lot of times you get silent mutations where this letter is different, but it codes for the same amino acid, or it codes for a similar amino acid. So be thinking of that as we are moving forward. The insertion or deletion is really the worst thing that can happen. You add something here, it's going to throw off the rest of the code. You take something away, it's going to throw off the rest of the code. Those are called frame shift mutations, and they're probably the worst. The worst is one that causes a disease. So if you insert or delete, you're going to get a frame shift mutation, and it's going to shift the whole code one way or the other. The insertion of 3 nucleotides causes an extra amino acid to be inserted during translation, and the integrity of the rest of the protein is still maintained. So you can add 3, and it'll shift everything over 1. Okay, let's look at another way that sometimes this chart is shown. So this is the common way. This is probably the chart I'll give you, but some people really like this way a little bit better for some reason. And it just starts you like, let's look for UAG, the start codon, right? UAG, right? Stop codon, right? Or you can do AUG, start codon. Do you see how that one works? So it's very similar. It's just a different way to look at it than the other chart. This is just the chart that I'm used to. Okay, so in addition to the codons that specify amino acids to a polypeptide chain, there are 64 codons to make protein synthesis. The triplets are all called nonsense codons or stop codons. That's these 3. So everything else codes for something, right? It codes for an amino acid, except UAA, UAG, UGA. I'm not going to make you memorize that. You'll have the chart. Don't worry. You will have the chart. I'm not going to make you memorize this chart. There's no point in doing that. But when you get to a UAA, it does not code for an amino acid. So it's called a nonsense codon. It's not that it doesn't make sense. It's just a stop codon. I tend to call it a stop codon. But you will also hear it called a nonsense codon because it does not code for an amino acid. AUG is the start codon. It codes for methionine because that starts when we talk about the ribosome and the tRNA. You will see that methionine is necessary. But the methionine there is what is the start codon. Another vocabulary word, reading frame. So the reading frame for translation is set by the AUG codon. So this will get you on tests or whatever. I will give you a long string of mRNA and I will say code for the amino acid chain. And you will just, if you just mark off 3, it's going to be wrong. You have to move along with your little finger or your little pencil and wait until you get to an AUG, mark that off as a start codon, methionine, and then go from there. And then when you get to a stop codon, you stop. So that is considered the reading frame. And the reading frame doesn't start until you get to an AUG. Okay, it's usually, and it is the 5' end of the mRNA. Remember, we counted our carbons, it's the 5' carbon. And that reading frame starts with AUG, ends with a stop codon. The arrangement of the coding table reveals the structure. There are 16 blocks. And again, you can also use that wheel if you like that better. I will probably give you both of them. And when you look at it, the reason I like this is because it just makes that degenerate code a little clearer, right? You can see that this is all going to code for arginine. So that's why I tend to use that one. Some blocks are divided in half, right? Leucine, all you need is the first 2. But if you go down here to serine, 2 and 2, some blocks are divided into half, ends with a U or C, and a purine, which ends with A or G. So you can kind of play with that, and we'll maybe talk a little bit about it in class as we play with this code. The specifics of amino acid, again, is that degeneracy. It's not an insult. It's believed to be a cellular mechanism, and it looks for it really reduces the amount of negative impacts from random mutations, right? Because all you need here is CUU and you're not going to get random mutations. Codons that specify the same amino acid typically only differ by one nucleotide. In addition, the amino acid with chemically similar side chains are encoded by similar codons. So aspartate and glutamate, which occupy the GA block over here, GA block, are both negatively charged. So when it folds, when that amino acid folds into a protein, it's going to act similarly, which will reduce the effects of the mutation. This nuance of the genetic code ensures that a single nucleotide substitution mutation, we're going to have some definitions, we're probably going to make cards of all the types of mutations, might specify the same amino acid anyway, which would be silent, or a very similar amino acid. So it wouldn't affect the protein product as much. Okay. Again, the genetic code is nearly universal. There's, of course, some weird viruses, and there's always, I think in biology, this, hey, we found this weird organism that doesn't use the genetic code. But by and large, 99.9% of things use this genetic code, which is fascinating, right? Like bacteria, cows, humans, plants, all the same genetic code. Just absolutely mind-boggling. Same consistence, mRNA, transcription, translation. There's only one genetic code that shares all of the common origin of life, which is just crazy if you think about the fact that we all share the same code. Okay, so just review. Can you look at the alphabet ATCG? Can you see how AUCG will be the same? We're going to do this in class where I give you a code and you tell me what the mRNA is and what the translated amino acids will be. And that is the genetic code. That is the that is, there we go, the central dogma. So if you can explain the central dogma, that is what it is. DNA to mRNA, mRNA to amino acids to proteins. That is the genetic code. So I will see you for the next lecture.